Hi, everybody, and welcome to Friday Night Flock Talk. I'm Robin Sullivan from the Leather Elves, and I'm here with Jack Pine from High Redbird. Hey, Jack, how are you?
I'm doing all right. How are you doing? Long time no see.
I know this feels much better. it, it was kind of weird sitting next to you last week. It was like, okay. It was
I know
pleasant, though.
I have to watch what I say when we're in person because if you want to, you can reach over and hit me. So,
You're
um,
really lucky that I didn't, so I'm just saying. Uh, So I hope everybody's doing well. We really enjoyed being live at AFA last week. It was a lot of fun to see some of you in person and to still interact with those of you that couldn't be there. So we promised you. See, look at how much fun we had. We promised you guys. I don't know, Jack. Is that your new friend?
I, yeah, I mean,
There were so many.
the AFA conference was fun.
It was a lot of fun, and we learned a ton. And so we told you guys we were going to do a recap, but honestly, there was too much to recap in one night. So what Jack and I talked about it a little bit, and what we decided was we would take topics and kind of work with some of the speakers that were at AFA, and over the next, month or so, or couple of months, bring some of them to you here on Friday Night Flock Talk. So, Jack, do we have any reminders?
Yeah, so I just want to let everyone know that this topic I am incredibly excited about. This was actually one of the first sessions in the morning, and I got up early specifically for this topic, and I was thrilled that I did. So, if you guys have seen the promos, you know what the topic is. It's avian genomics. If you don't know what that is, we're going to go over that. But before we get into that, first and foremost, we always encourage you guys. You want to make sure that you're following Robin and I on social media. We do these live streams every single week at Friday at 7 p.m. Eastern on Friday nights from the Leather Elves Facebook page. So if you make sure that you like the Leather Elves Facebook page, you will get a notification whenever these live streams start. That way you don't miss out on any of the fun that we're going to have. Now, of course, we do post these live streams in a couple of other places as well. If you end up missing them, you can always watch them on the High Redbird YouTube channel. Subscribe there. You can watch all of the videos. They are all put together in one playlist. There are over 40 hours of content in that playlist. Robin and I are closing in on a year of having done these live streams every single week with you guys. I know. How did we get here?
It's kind of exciting because it's taken on a life of its own, and I really do enjoy the the community that's been built and the group of folks that come every single week, and we keep adding every week. So don't forget to bring your friends too.
Yes. And we try to give you so many different ways that you guys can see what is coming up. If you guys pay attention to the Leather Elves Facebook page, the High Redbird group on Facebook, we'll post notifications of what events are coming up. But then we also have both of our Instagrams, either the Leather Elves or High Redbird on Instagram. You'll get notifications about topics that are coming up, any fun things that we're doing. But also, Robin posts a lot of fun pictures of different toys, different types of animal enrichment. I post a lot of photos of baby animals because that seems to be what pretty much runs my life. Um, so, a whole lot of fun that you guys don't want to miss out on.
All right. So we have a special guest this week. And like Jack said, this was one of the sessions. This gentleman did one of the sessions at AFA. It was an early morning session, and he still had people go. So that tells you it was kind of a exciting topic. So it's my pleasure to introduce you guys to Taylor Hains. Taylor is the founder of Terra Wildlife Genomics. And rather than read your bio, Taylor, I think it's better if we just kind of jump into things. So welcome, first of all.
Thank you.
And so
Thank you for having me.
maybe, oh, it's our pleasure. So maybe you could tell us a little bit about you. So where did you go to school?
So I went to undergrad at Columbia University.
I graduated with a degree in environmental biology, which don't ask me what that is. The faculty didn't even know. And so then I moved from New York City. It was actually really funny. Like when I was trying to pick programs, like I literally kept going, "What is environmental biology?" To the director of undergraduate studies. He's all like, "We just have a name. That's all I know." Basically, it's ecology and evolution. So I moved from New York to DC, and then I enrolled in at Johns Hopkins University in an online program in environmental science and policy. But I worked as a contractor, and then I did my master's thesis work at the National Zoo and the Natural History Museum. And now I'm here. I'm here in Chicago at the University of Chicago in the Committee on Evolutionary Biology, working at the Field Museum on parrot chromosome evolution and conservation genomics.
So that sounds like a lot. It's a mouthful. So before you explain that to me anyway, and I'm sure there are others that need that explained, why birds? Why are the birds the focus of your study?
I think I'd like to thank my grandmother. She got me a budgerigard when I was very, very little. Sadly, and may his soul rest in peace, we did not know how to take care of him properly. But it just got me enthralled in bird conservation and also telling new pet owners how to take care of a bird properly. Me and my family did not know that parrots eat the inside of the seed and leave the husk, so we thought he wasn't eating. And we learned the hard way because no one bothered to explain it to us. And literally, when I was growing up, I was the person that my aunts and uncles and my neighbors would call, going, "Taylor, there's this bird I don't recognize. What is it?" Of course, the weirdest one was my neighbor calling, "Taylor, there's birds that don't look like they belong here." And I'm like, "Well, what do you mean?" So come over. And inside of the top of a rat cage, they had two lovebirds that have flown into the garage. And I'm all, "Yeah, lovebirds don't live in Southern California."
No. So, well, and
Well,
you know,
and
I
I
think we
love.
all have.
I was gonna say
Go ahead,
I
I
Jack.
love
cannot
hearing.
remember what
I love hearing that you were looking for more information on how to care for birds and how to, me and Robin are lightly biased. We have a horse in that race because we have been working to educate people on the care of their birds. So hearing that people like to do those things, awesome for us.
well, so one of the speakers at the at AFA conference, I love the phrase she used. These birds are in our charge. I truly believe that we took these birds in from the wild, and we are responsible for their health and well-being and for their happiness. And so, I mean, my birds are my best friend. I had a bird pass away last year, and everyone checked in on me because they knew how much I loved that stupid bird. He was my bubby, and I'm gonna start tearing up. It's kind of, I don't know if it's kind of sad that like basically my closest relationship is with birds, but you know what? I'm just gonna go with
No, I think we've all had, the budgie growing up that, it was, I remember at my Nana's house, we had a budgie that lived in the dining room. And that was when everybody smoked, after dinner. And there's the little budgie, and nobody knew that was bad, and but he lived a decent life, so, but we get better as we go on, we evolve in our care. And I think the work that you're doing is something that is part of that evolutionary program. It's part of that plan. And for companion parrot owners to find out more about that is really exciting to myself, and I know it's exciting to Jack too. So, Taylor, what is Terra Wildlife Genomics?
So it's a new nonprofit. It's relatively new, so it's just me by myself. Um, and so, of course, trying to manage everything has been difficult, but we've been doing pretty good so far. It's a nonprofit that has four main goals. The primary one, and it's the one I have the most passion for, I always wanted to breed some sort of animals, primarily birds. I tried to breed budgie guards at one point, could never get them to mate, actually. Zebra finches, however, they just mate whenever it rains. So I had a flock by the end of 1 year. But it is to manage captive breeding programs using genomics and genetic data to create healthy and sustainable populations that can be reintroduced into the wild. It's also to create genomic tools for helping to enforce wildlife trafficking. Because genomics is an amazing toolbox that I find it a big puzzle, and you can use it for so many different things. You can help it to manage a population. You can help it to protect the population. You can help it to do something in policy. Like there's a lot of it's a lot of my family is artistic. We have musicians, we have actual artists in my family, and I consider my art to be science, particularly genomics. Like I just somehow put together these things. Um, and then it's to manage populations in the wild in real time and scale. That's one big thing, especially when it comes to like important species like fish. But then also to increase diversity in science. As a young black gay man in science, I am basically the only one. I am lonely by myself, and I've gotten very used to that. But I want to bring more people that look like me and act like me and talk like me into science and conservation. Doesn't matter what you want to do, whether or not you want to work on fish, whether or not you want to work on birds, whether or not you want to work on humans. I find humans boring and fun. But it's all the same, and we all have the same roots, and we're all part of the same community. So
Very cool.
Yeah.
So, Jack, do you have, I think we Jack and I have discussed, what we want to talk to you about, and we did the outline that we always do. But I think we need to start at the very beginning. Would you agree, Jack? Oh,
Ready.
Taylor, we don't have smart glasses. We have to put on our smart
No,
glasses.
Taylor has his built on. Yeah.
He has it. I don't have any. So, Jack, you'll have to
I
ask the question.
without them.
Well,
I don't know if I had that at the conference, but most of the time I could not see at the conference, actually.
So
that would explain your choice of some of the people you talk to.
Oh, that was mean. That was mean. I think you're my non-sexual life partner right now.
Nice. Oh, by the way, Taylor, when Nick thinks we've gone off topic, we get butterflies, just so you know.
Okay.
Jack,
okay, we'll go back on
question.
topic.
Did you have a question for Taylor?
No. So for this evening, we actually wanted the people watching to sort of, guide the conversation, because we don't want to go back to, a middle school understanding of things if people are ready for, a in-depth analytical understanding of this. But at the same time, if anyone has any questions that they want completely broken down, please feel free to put them into the chat, because we definitely want to address all of those.
For that.
I think we definitely want to start with, I guess, what is a genome? That seemed like a good place to start. And realistically, I think we've seen several Jurassic Park movies that might leave people concerned about us studying a genome. So what is it?
Let's just say I have a drinking game. I drink to every scientific inaccuracy in Jurassic Park and Jurassic World, and I get drunk within 5 minutes. When the first Jurassic World came out, I was living in New York, and MTV interviewed a PhD student at the American Museum of Natural History, and they asked, "How do these hybrids get formed?" And his answer, I just kind of almost wanted to text him, just like this: "You are a paleontologist, not a geneticist. Don't answer these questions." A genome it's complicated. So genomics refers to nucleotides. So basically, hopefully we all know from high school, middle school biology that basically a DNA sequence is made up of 4 base pairs: A, T, G, and C. So when we analyze, anything in the genomic content, we're looking at those 4 base pairs, and those 4 base pairs will span across chromosomes, and there are multiple chromosomes, obviously. So when you have a reference genome, so the human reference genome is every sequence should equal a chromosome. That is the ideal. Of course, with humans, there's a lot of medical reasons why we want to study the genome. So there's a lot of more money behind it, so it gets done more quickly. We literally, not too long ago, just completed the human genome. It's complete. This is not necessarily as easy and the same for birds or reptiles or amphibians. And then, of course, you have RNA, which is which is called the transcriptome. That's a totally different thing. Proteins, that's a proteome. It's a totally different thing. But a genome is those 4 nucleotide bases spanning across chromosomes. And of course, there's going to be proteins in there that make the center of the chromosome, the ends of the chromosome. So that's the definition of a genome. So when they've done a genomic sequence and they looked at the variants, they're looking at the differences between the reference and an individual at those particular sites. So if in the reference, if it's an A if it's an A and it's actually a G in something, in let's say it's an A in me, it's a Jack, in Jack it's a T, and in Robin it's a C. Those are variants, and those variants can affect skin color, for example, or hair color, even though I know that's not Jack's real hair color.
It's my natural color.
Okay. Okay.
Blue.
A red like that, probably not actual real. If it was real, Jack, we need to have you sequence.
We need Jack's genome.
You can fight me for it. So basically, it sounds like we're just mapping out DNA and then trying to find differences. Would that be the simplest?
That's basically what we do. We try and find differences in every different aspect of the genome, even of cells, even of organs. That's basically most of what I do.
So what kind of things can we learn from studying genomes?
So there's a lot of things that we can study in relationship to birds. There's also a lot. Just because, so basically, we have model organisms and we have non-model. I love model organisms because they are a very difficult puzzle. It's a lot more difficult. You need to put the pieces together. Human and model organisms, would be considered mice or yeast or fungi, something that you can easily grow in a lab and perform experiments on. And because of the principles that we learned from model organisms, we can do a lot in non-model organisms. So when it comes to birds and it comes to bird conservation, we can figure out if you have sorry, my bird is walking in the background right now for some strange reason. That we can learn if you have a reference panel. So if you have samples that you know where they came from, you can figure out where the birds initially came from. So for the blue-headed macaws, the blue-throated macaws, the golden conures, if we have samples from across their wild distribution, we can use that to basically figure out where the birds that originally founded these captive populations we have as pets, and figure out where their ancestors came from. It's basically the same principle that Ancestry DNA and 2-3andMe does. I honestly, as a geneticist, think they could explain the results of Ancestry DNA and 2-3andMe better. But you know what? They have their own thing. So I've even thought about doing an Ancestry DNA for your bird.
You should.
Those are possible.
I can tell you, my son and daughter-in-law just did the DNA on their dogs. So if they did them on their, and they, it was like, oh wow, this new puppy's going to be huge because he has Great Pyrenees, blah blah blah. She has Great Pyrenees in her, and I said we probably didn't know that before you adopted her, but good job. And but I mean for a bird, it would be super cool. I think people would be interested in something like that.
No, and it's incredibly important for conservation because And the one big thing that when I started Terra Wildlife Genomics was basically, okay, so we'll take an example of, and I think Nick can show actually the next slide I have, which basically has the AZA birds that are in AZA population. So AZA stands for the Association of Zoos and Aquariums. So basically, all zoos and aquariums, with their grain and salt, are either part of this association or affiliated to the association. Basically, the association has particular criteria that are there for the well-being of the animals and for conservation. Like random example, whale tank size requirements. We all know that's a hot topic issue, but the federal size regulations for whale tank size is lower federally than it is for AZA. To receive AZA accreditation, you have to have a much larger tank, which associates with better health of the animal. And so these are the 8 species in the U.S. of parrots that have captive breeding programs. And some of them at root people should recognize from pets, hyacinth macaws, for example. So, Robin, Jack, do you guys have a hyacinth macaw by chance?
I do not, sadly.
I wish.
Okay, do you have a
Do you have
golden conure?
extra?
Okay, do you have any of these species on the channel? Okay, so we're going to move on from that point. But there are people that at AFA that have hyacinth macaws, that have golden conures, that have palm cockatoos. And so what we can do with the baby, stop it.
We can help you with
having
that.
a bird is like having a child. Having a bird is like having a child, and he wants attention right now, and I can't give it to him. So, but we can use the genetics for these birds that we have in captivity, so these pet birds, and we can figure out either where they came from, or we can try and figure out whom. I should really cover him right now, but I think he sees birds on the screen, and he's just like, no, no, Dad cannot hang out with any other bird but me. I love you, but I will cook you.
We've all been there, so trust me.
Butterflies. So you can use genomics to basically figure out pedigrees, figure out genetic relationships, figure out genetic health of the organism. You can even look at genetic traits. So there's a lot of different things that we can figure out from this, from genetics, and even proteomics.
Go ahead, Jack.
No, and I love how you're bringing up the AZA, the Association of Zoos and Aquariums, because I think for a lot of people, they don't realize what AZA guidelines entail for pretty much any type of permitting that you may need, be it to give you guys an example, for me here in Texas, there are things that require USDA permitting. There are things that require Texas Parks and Wildlife permitting. There are things that require federal permitting. For most of those permits, there is going to be a list of criteria you have to meet or be AZA accredited, because those organizations realize that AZA criteria are typically set higher than just about everything else. So especially that is definitely the standard for care for animals. It's going to be optimized, enclosure size, optimized diet, optimized enrichment schedules. So I think it's great to bring that up and bring up the work that they're doing with all of those different species.
Oh, definitely. And I think too, go ahead, the species survival plan.
No. Yes. Growing up, I wanted to be a population manager for a species survival plan. I'm going to get up and cover him real quick. I am so
sorry. So just for anybody that's unaware, the species survival plan is it's a way that there's a person that has the stud book, and they're the person that tells you, okay, you have bird X at your facility or animal X at your facility. You have this person has animal Y. Genetically, they are a good match. this is where they need to be, and that's how animals are put in breeding programs for zoos. I mean, that's the very basic description of it. So Taylor, you're working with the SSP?
I'm working with trying to put together a couple different SSPs and also make new SSPs. Because again, like we said, like there's only 8 parrot species SSPs. There's 131 recognized species right now. 1/3 of them are listed as endangered or threatened. And so like there's species on that list that really should be included, like the gray-breasted conure, or the actual official common name is gray-breasted parakeet, has about 350 to 400 individuals left in the wild. They live on one small mountain top. Potentially, you could go find and buy them in the pet trade. And so the whole thing about Terra Wildlife Genomics is basically putting together, and so I believe basically there's different associations across the world. So AZA is the U.S. Association of Zoos and Aquariums, but like in Europe, they have the European Association of Zoos and Aquariums, the IAZA. And so they have a gray-breasted conure stud book. Also, they have a blue-throated conure stud book. Anyone doesn't know what a blue-throated conure is? You really should. They're gorgeous. They're my favorite. I want one, but I know I probably should not have one. And so there comes that conflict. But the sad thing is about stud books most of the time, and that I've learned in the past couple of years, is they say there's genetics, but they don't include genetics. They base their genetics on pedigree. So there's no actual genome sequencing or genetic sequencing of any sort. And so when it comes to captive breeders, it's the same kind of issue. There's just kind of maybe a bit of a higher scale in care, but maybe not really. It's kind of like it's ambiguous. So a breeder has a thought of like basically one of the big issues with a lot of endangered species is that someone goes out and collects a bunch of species before, back in the early 1900s, 1950s or something like that. And they collect a few at this one site and they go a couple hundred, a couple, maybe tens of miles, 20 miles, 30 miles away. And they go to another site and they bring them into captivity and they assume that these animals are unrelated, which of course we all move away from home. Some of us don't want to be like, we love our parents, but we don't want to be near our parents. The same is with some animals. And so they assume unrelatedness, and that sometimes is not correct. Sometimes it all depends on the species. Every species has different biology, and so the founding populations of any of these parrot SSPs is some of them are actually quite high, but some of them are quite low. And so the goal of Terra Wildlife Genomics is actually include agriculture and captive breeders and maybe even pet owners into these captive populations. So it might be like, hey, breeder, trade this one bird with the zoo, or this private owner that you have one. I mean, the story of the Spix's macaw. I'm hoping most of us saw the movie Rio. I watched it on loop. Like, I actually watched it on loop. I have the whole movie memorized.
I want to see.
I met Anne Hathaway. Yeah, if I met Anne Hathaway, I would basically recite all her lines to her from that movie. I love that movie so much. Also, it's the reason why I'm afraid of cockatoos, because Nigel the cockatoo is evil. That's really the only reason why. They're not bad. I mean, I held the palm cockatoos at the conference, and you don't know how much I wanted to pee my pants.
Well, and we did have someone who asked, and you may know the answer to this. Someone asked why the palm cockatoo and the hawk-headed parrot are included as species survival plan birds if they are not threatened in the wild.
So it's threatened or endangered. So basically, you have multiple criteria. So like basically, there's this 3-tier system for threatened. And then you have endangered, and then you have critically endangered. And it depends on particular criteria. Now, also ACA is potentially considering throwing out stop with most parrot breeding at zoos because they're finding it hard to either find the birds or find technical expertise into breeding these birds at a high impact or high reproductive rate. Which I literally saw that report and I wanted to drink because I'm only because agriculture is where you go. Agriculture, like again with that example I gave you guys before, there's a family-owned business where I grew up, and they were breeding this endangered species of dove. And they went to a conference, and the zoos were going, "We can't get these birds to breed," and they were going like, "Well, we just have a flock of 20 of them from like 1 year alone. Like, I don't know what you guys do." But again, in the defense of zoos, one of their biggest issues, two biggest issues, is number one, space. They don't have enough space for all the animals, especially when you think of a metropolitan zoo. And then number two, money. The reason why the San Diego Zoo is so expensive is because they use those funds not to make a profit, but to care for the animals, pay for the staff, and pay for on-site conservation. Which, coupled with what I do, we need to preserve intact habitat for all these species because otherwise, captive breeding programs are useless because we can't keep all these animals in captivity for the rest of eternity. So the one thing I can plead to everyone watching this is, you know, go to your local zoo, support your local zoo, support your wildlife sanctuaries, support your breeders, like the enroll in conservation. And when you see those really sad WWF commercials, the World Wildlife Fund about rhinos and elephants, supporting those has saved other species. We call those keystone species. I mean, I'm going to be very sorry to say the panda would go extinct without it. Apparently, pandas reproduce by know how to reproduce by watching adult pandas mate. So in captivity, when an adult to see that from, they have to figure it out. That's not necessarily the best life strategy. But you can fundraise so much because people are going to want to save the panda. But then you also save multiple other species that live in that area, which might be a parrot, might be some passerine bird that is popular in the pet trade. It could be anything. So that's the one thing about all these organizations, including agriculture, because I know so many bird breeders that do conservation work as well.
And I think too, Taylor, I have found I've met people while I've been on the road doing sales and things and doing education pieces. I've met people that say, "Oh, I have a such and such, this bird," and I kind of go, "Wait, what?" I ran into a woman that said she had a pair of greater Vossas. Excuse me? I mean, you don't find greater Vossas in the pet trade all that often. And she said they had a pair and she got them back in the 70s. And, she wanted only one, but the guy would only sell her two. And they produce. And I'm like, okay, I need to hook you up with people who are studying the Vossas, and it's just.
Thank you. I mean, to me, that just, and sadly, I mean, I think I'm single because I have a passion for this because I get really upset about moments like that. And not that she did anything wrong or she's doing anything wrong. It's just she doesn't have the information to go, okay, what do you do with those birds produced? Like, no one has it. If you're able to produce it, write the information down so other people can do it. Like, take down the records. Like, I mean, thank God I only have one bird because if I had, a pair and they started reproducing, I would basically have a small little stud book in front of me right now that was just by me.
No,
And so, like, one of the things from my AFA conference is I'm going to be turning that into a stud book.
Very cool. And, and I think that's important. Jack and I talk to everybody, every single week. We talk about record your information, write it down, videotape your, training sessions or what you're doing, and then share it with other bird owners, because it's keeping those records. At some point in time, you guys, Taylor might call you and ask for this information. So you guys need to write this down.
Yeah, I mean, it's going to happen sooner than later.
Yeah, there we go. So we've talked about AZA, we've talked about the SSP. So how does this happen, Taylor? How do you get the information that you need?
So basically, and one of my collaborators actually calls me the Facebook biologist because I'm part of the Conure Owners Unite Facebook group. and I've seen very interesting, very cool birds, and I'm all like, ooh, I know what species that is. I don't have a sample. And so basically you can use a variety of different things from a bird. So ideally, I love to work with blood. If people are comfortable with taking when they take their bird, and I say this in the sense of we call it opportunistic sampling, where when you take your bird into the vet, ask the vet to bleed the bird. Don't specifically take the bird in to be bled because you don't want to cause the bird extra stress or extra harm. So just when it happens to happen, where it's in a veterinary situation, or if the bird happens to cut itself and it's bleeding, try and take a sample. That's opportunistic sampling. I remember when I got to the Smithsonian for my first fellowship, I was working on crocodiles, and so I asked the director of the genomics lab, like, "Hey, I'd like to get a sample from the Cuban crocodiles at the zoo." And they said, "Oh, we will submit a opportunistic request, but we don't think it's going to happen." 2 weeks later, apparently two of them got into a fight. One of them broke off a jaw, and so they had it in the veterinary hospital. So he looked at me going, "Wow, I didn't think that was actually going to happen. But here's your sample." And so blood, because birds are also nucleated, so their cells have nuclei in it. For mammals, we don't have nuclei, which basically the nuclei has all the chromosomes, all the DNA. So blood is preferred, but if a bird drops a feather, that works as well. That works almost just as well. My baby dropped finally after almost 4 months, dropped 3 feathers. And so right now his feathers are digesting so I can do an extraction. So I can figure out whether or not he is a he or a she. I still don't know. And so you start with those, and then you do what we call a library prep or PCR and sequence parts of the genome or the whole genome, and then you analyze. That's, I guess, the base sum of where the information comes from. And if there's a reference genome, which a lot of what I've been doing is generating reference genomes for different species that aren't available or aren't published. So the dusky conure reference genome, the crimson belly conure reference genome, and the green cheek conure reference genome are all that if you go onto NCBI and look up those genomes, those are all my personal birds. Because I'm the crazy guy that when I take my bird to the vet, I ask for a blood sample, and they remember me even years later. And now I'm the Facebook biologist, and I've gotten actually a lot of different birds from Facebook because people are willing to contribute to this research and find out information about their birds.
Well, and so you mentioned your bird having those feathers, doing that testing to determine gender. So how does that determination of gender works with birds? How does gender differ for birds?
So gender, okay. So I hope we all know from high school biology that Jack, you have an X chromosome and you have a Y chromosome. Robin, you have two X chromosomes. Now in birds, it's reversed, and they're not called X and Y; they're called Z and W. I'm still not quite sure exactly why that is the case, but male birds have two Z chromosomes, and females have a Z and a W. So hypothetically, if anyone wants to send me a sample of their bird, it should be a reference genome. Ideally, it should be a female, so we get both that Z and W. But when you send out for genetic sexing from a pet store, from a vet, basically what they're doing is amplifying a very small region on the Z chromosome that differs in size very slightly, like by a couple base pairs between males and females. And so males show up as one band, or as two, one band, and females show up as two, because on the Z and the W, they're different sizes. The chromosomes themselves are different sizes, but the gene region itself is a different size. And so basically, if you're looking through basically what we call a gel electrophoresis gel image, you see two bands that are relatively very close together, and then one band. Two bands is a female, the one band is a male. That is the standardized technique for genetic sexing. So whenever you get your bird genetically sexed, hell, I could do it. I mean, I've genetically sexed all but one of my own birds. And that's the only reason I did not do it the lab work myself was because of COVID, and I didn't have access to a lab. So, like you can see, you have the X and Y, and then the Z and W. Those chromosomes are different size, and so that gene region is going to be different sizes on each of those chromosomes. While on the Z chromosome, it's going to be the same. You can do the same analysis when it comes to humans. It's across the board. The only organisms that differ is when they don't have sex chromosomes. Like crocodiles, alligators, and some turtles don't have sex chromosomes, so you can't do this type of analysis.
It's that Jurassic Park thing again, isn't it?
I kind of want to do Jurassic Park in real life. Like, I really do. But I need genetic material from a dinosaur.
Well, I mean, the other
so
thing there.
Yes.
Oh no, I was just going to say, you could do that for humans. The difference is that we look different. This is a bigger deal for birds because they're not sexually dimorphic, not sexually dichromatic. Most of them.
Yes. I mean, for parrots, I believe there are 3 species. The one, the eclectic parrot. Second, budgerigards. And I cannot remember off the top of my head the third one. I mean, even my advisor didn't know that budgerigards, the males after maturity have blue noses and the females have a reddish nose. And these are like things that can be common, but also like misappreciated, like unappreciated. Just the subtleties. Like budgerigards would almost be considered a model system for parrots.
Are there any species, Taylor, that you are particularly interested in that people could help out with, or
any Pyrrhura species? So blue-throated conures, gray-breasted conures, green-cheek conures, crimson-belly conures, pearly conures, maroon belly, basically all those ones that look like. Also, macaw hybrids. Macaw hybrids are potentially some of the coolest genetically and chromosomally. I say potentially because I haven't, this is my PhD dissertation. But there's a lot. I say messed up, but I say messed up in a good way. They're messed up chromosomally. Like, they're messed up. Like, it's a fantastic puzzle piece that I'm just like, everyone that I've shown, like, preliminary data from other papers, they just look at it and just go, okay, so Melissa just wrote cockatiels maybe. Cockatiels can also be fascinating and model organisms as well, considering the fact there are so many. But chromosomally, and that's what my dissertation is going to be on, macaws are the jam. Also, green cheek conures and all those relatives. So I'm biased. I'm really biased. But even color mutations are fantastically interesting. Maybe not on a chromosomal level, but on a genetic level of just figuring out what genes work for, like what genes cause. Like with budgerigards, all the color mutations that we see, almost all of them, we know the generalized mechanism by which that happens. So, there's not enough melanin in the feather, so that's why they look one way. Or there's not enough carotenoid in the feather, and so identifying those genes, and we all potentially know that there are what makes me different from Jack, different from you, Robin, is we have a bunch of different genes affecting the same thing. So all of our skin tones are different. And that's because there's multiple genes acting upon it. And so the same thing applies with birds. And so trying to figure that out. So if, green cheek color mutation, if people want to send me green cheek, like a bunch of, green cheek color mutations along with, the normal type, we can figure out exactly what genes affect those color mutations. How do we get the turquoise color? How do we get the high red factor coloration? Which I'm confounded that that's even a thing. Potentially also, is it a fact that they hybridize because apparently I've seen in the conure Facebook people say, "Oh, I have a green cheek hybrid with a sun conure hybrid," and those are two different completely genera. Those are a minimum of 6 million years divergence. How does that happen? Those are the questions that I would love to answer. And then there's also conservation implications and management implications. So potentially knowing how that happens between a green cheek and a sun conure can tell us about the genetic health of that individual. So what, potentially maybe what you have to watch. Okay, the mechanism that separates the two is there's going to be a heart issue that causes most of them to die before their reproductive age. I'm just making this up right now. This is an example. And so potentially when breeders breed these birds, they can go, oh, this bird is going to have heart problems. So we're going to watch the heart particularly. Does that make sense?
So, I mean, and so that's an application that can really help. It has the potential to help companion parrot owners as well, in that if there's that predisposition to a particular health issue. And is that something, Taylor? Go ahead.
No, and that's something I've been really interested in because I had a green cheek conure for 3 years. His name was Orion. He was the love of my life. And I am not joking. He was the love of my life, and he passed away at random. It was horrifying, and it was at the beginning of the pandemic. And the only thing I can think about, because I didn't change anything in what I was feeding him, there was nothing new and special around him, so I don't think he ate something foreign. Like, I could not figure it out. And so one of my initial thoughts was maybe the green cheek population and is inbred or genetically unhealthy, or there's this goes on in particular lineages that we can't necessarily see until it's too late. Like, he was 3 years old. Ideally, if I was raising him correctly, obviously he should live till 20-ish. And that's not the fault of breeders necessarily, but because one doesn't have the information. Like, I only found out that for blue-throated conures from someone I met at the conference that all the blue-throated conures in the U.S. are descended from 1 to 3 pairs 20 years ago, and so they're highly inbred. So the chicks are kind of wonky when they're born. This is the importance of agriculture and having this community that people know these things. And so I'm let's try and figure out what we can do to fix those problems with what we have now.
Well, and you were talking about this earlier when you're setting up a stud book, when you're setting up what pairs should be breeding, talking about founders, those wild caught animals that are typically seen as being the most genetically diverse from everyone else. But if we get them all from the same area and not knowingly put together full siblings saying that they are genetically diverse because they're from the wild, but they're not actually, as genetically stable as birds that we already have in captivity, that can definitely lead to problems. So I think understanding the genomic breakdown of different species, different individuals especially, just is so incredibly helpful for the long-term health of aviculture.
No, exactly. And actually, I'm looking at the chat and question Brian asked. I did my undergraduate thesis on hybridization between scarlet macaws. And before my thesis, there wasn't actually any confirmation that scarlet macaw subspecies could hybridize. So we don't know about what, and some people at AFA told me that, they believe that most of the scarlet macaws in captivity are hybrids between the two subspecies. Also, we didn't necessarily know where the species boundary was between those two subspecies. They thought it was the most political north boundary of Costa Rica, when in actuality it's the mountain range that runs through the country, which makes more sense, obviously. But captive breeding, birds don't care about human politics, which is why I love birds, especially with these past 4 years. Why I love birds. Why they don't care.
And the nice thing is too, if we're looking at these trends, these propensity for a specific not defect is not the word if there's you're going to see this in a population. And if by diversifying that population, you can alleviate that. Because like you said, Taylor, breeders don't always know, because it may not occur till, 5 years from when they hatch out. So you don't know, and the breeder's not going to call up everybody and say, hey, how's the bird doing 5 years later? So it's really important. And I think, you talk about politics, and in the bird world, in aviculture, we run into our own sort of politics where it's the rescues and the breeders and the companion parrot owners and the big flock breeders and the hobby breeders. And what happens is there are these little pockets of information. And I think this is a great argument for really working together and trying to, so because there is that information out there. And I think sharing that, can lead to a more diverse population, genetically diverse population, and a population that may in the end be healthier because they're coming from the information's coming from different places.
No, exactly. And one of the things I hope by going to the AFA conference and by talking with you guys is that people would reach out and go like, hey, I want to participate. I want to donate some of my samples. And while I can never tell a breeder, and I will make this very concrete, I can never tell a breeder what to do with their birds. I can make recommendations, and you can follow them or you cannot, and that is up to you. That's primarily how zoos mostly work. Because they're part of AZA, there is some kind of, if you don't transfer an animal, the population biologists say that you're going to, like, that is up to the zoo, but there might be some fallback. With captive breeders, of course, these birds are privately owned, and I cannot and I will not take someone's birds because they don't do what I say. I can tell them what I think they should do, and I can give them the information to decide for themselves. But I believe, and I wholeheartedly believe this, that we, as a community, zoos, private breeders, private pet owners, can save these species if we work together and if we talk to each other.
Mm-hmm.
I'm all about communication. So, if I said something that offends you during this, time we've been on, please tell me, and I will apologize profusely, and I will make sure that it doesn't happen again. It's that kind of communication that my parents and my family taught me was incredibly important. That I think was one of the reasons why I've gotten to this point in my life, and I want to use that to basically try and save these species because, I am so thankful for having my bird, especially through the pandemic, because I would not have been able to keep my sanity intact without him. And so I want to either, help him and help his species survive, despite all the mistakes humans make, because we are a very, very stupid race.
I know, I can tell you, Taylor, I know we've got a couple of folks who breed blue crown conures that are on the flock talk on a regular basis, and maybe you could connect with them. And I think it's there are a lot of different personalities in this group, and if you've offended them, Jack and I offend them on a weekly basis, so not to worry.
It's all a difference of whether or not you talk about it and go, hey, you've offended me, let me apologize. But I think I've sequenced, or it will be sequenced soon, a reference genome for the blue crowned conure. So I've all the neotropical, all the conures and macaws, almost all of them I've gotten so far. So by the end of this year, knock on wood, we'll have those done, so we can use those as references to basically use as a panel to map a bunch. If anyone wants to send me their blue crowns, please do. I'll sequence it. I'll let you know what sex the bird is, where it potentially came from. We'll do some work with museum specimens, which is also a big thing. Museum specimens are basically our reference panel of what population structure looked like in the wild before we humans decide to mess things up.
Well, and so I just want to bring up, so this sounds incredible. We've seen the applications that zoos can use for determining mean kinship relationships for their birds. We've seen how private bird owners could potentially know about potential afflictions or genetic abnormalities that their birds are prone to. you can know where your bird comes from in the wild, which I think a lot of people would love to know. But I mean, I guess one question pretty much everyone would have is, so this is sounding a little bit expensive. So how much does this cost, and like is it getting cheaper?
Okay. Anyone that knows me or has worked with me so far knows I'm the cheapest person you will ever meet. And I have learned to do genomics on a budget. So getting, how is that off topic? Okay, you know what? I'm just gonna ignore that. And I'm gonna get back on topic. But so sequencing this stuff, it depends on what type of method you want. You can do whole genome sequencing, or you can do a reduced sequencing. So what I presented at the AFA conference, which is on blue-throated macaws, was a reduced genomic representation. And that can be incredibly cheap. It's getting cheaper. So it depends on what kind of questions you want to ask. Now, if you want more functional stuff like diseases, we need to do a whole genome approach, and that's more expensive. That would be about $300 per bird ish. I'm getting it down because I even just found a sequencing core that does sequencing for cheaper. So I'm like, yes. But when you do the reduced ones, it can be like dollars per sample, and you can still get usable information. So it depends on how much you want to do, what you want to do at what scale. So actually, to show you, like for pedigree analysis, so that the pedigree that I presented at AFA, that was done using a reduced representation library. Now, if actually we can show that real quick, essentially, if Nick. Okay. Well, eventually, if it shows up again.
There we go.
Yes. So basically, we have this Aviary A and Aviary B. So actually, we have one of these owners actually on this chat right now. And so basically, we have at least two families, maybe two and a half, because there's some cheating going on, at least in that. So basically, that top line is basically fake parents that the program assigned. And so for that Aviary A, the second line is the actual individuals that were actually sequenced, and then the offspring were sequenced. So we have some cheating going on. So it's kind of two families, kind of three-ish, maybe. And so then you have the second aviary, which they're all related. Now, for the first aviary, one individual was excluded. And for the second aviary, three individuals were excluded. And that potentially can be correlated to the fact that we use a reduced representation library versus the whole genome library. So you're not getting as many of those variants, nucleotide differences, that A versus C versus T versus G. And so really, you can do this at a very baseline for incredibly cheap. I'm surprised how much the prices have come down, and the prices continue to go down. And when I say cheap, it's within the hundreds to thousands, but it's not like tens of thousands. This project was done for a couple thousand, even I think maybe less than 1,000 per aviary was sequenced.
So you're saying that's for all of those birds that we're seeing, not one individual bird, but that's for all of the birds in an aviary.
Yeah.
Wow.
So basically, each aviary was 16 individuals, I believe, and so it was like about 1,000 for one, about 1,000 for others, so 2,000 across the board. And if you pool up with people, if you share resources, it gets even cheaper. So it's kind of like, it's again that community we're talking about, the information, the helping out. I cover this, you cover that, it brings it down cheaper. So genomics is not that expensive anymore, and it's continuously getting cheaper, which then apparently the sequencing companies will make no money, and then we'll be stuck with nothing because they won't do it anymore, probably. But it is getting drastically cheaper.
And and I think in general, this is something that not everybody's into science, but I think everybody's into better quality of life for their birds. And, and I think that comes through science. We've talked about, when you talk about what the origin of the kinship is, where does this bird, come from in the lineage? And I think that makes, for me, as someone whose passion is enrichment and husbandry, that's where I can say, okay, so this bird, its relatives come from X. And then I can say, okay, so let's create something that you might find at that place, or let's create an opportunity that mimics that location. And people always, when I ask people, where did this bird come from? I don't want to know it came from Joe's pet shop. I mean, that's important because you need to know that part of the history. But let's talk about the bigger picture. And I think that's something that can come out of this as well.
No, exactly. And I mean, I love how, what was it? One of my lab mates, I consider myself a bioinformatician. I like putting together the puzzle pieces on how to put together the genome and whatnot. And so he said, "Oh, I have this idea. Why don't we do this?" And I kind of just smiled, going like, I've taught you well. I'm a proud mom." And you just did that same moment where you're just like, "Hey, if we know where the birds come from, we can figure out what's in that actual environment and make enrichment." So that identifies the birds from that region. And it's kind of like, it's a puzzle piece. It's biology, and this is not that hard. I think people get scared off because we're very jargon based. And so I apologize for anyone that doesn't know the specific words I mean. But it's, once you understand the definition, basically you just have to learn the definition, and then you're pretty good to go.
And
So someone
I
just
think
asked whether or not habitat.
No, go right ahead.
Yeah. Sorry for interrupting. There's a question that's like, is habitat destruction a greater threat to wild parrots than bird trade? I think it's kind of mixed, honestly, because we don't have a place to reintroduce the birds in, and their habitats are either getting more restricted. And then you have the illegal pet trade. I say the illegal pet trade because, I mean, and I honestly don't believe that we need to bring any more of the species that we have in captivity more in to maintain genetic diversity. As long as we use the tools and the knowledge that we have, I think we're pretty good. Of course, that might be different if there's a species that only has 2 individuals in captivity, and I just don't know about that. But that also relates with how we treat other people. People that live in impoverished areas that don't have enough, their jobs don't pay enough, are more likely to, participate in wildlife trafficking, whether or not it's hunting elephants and getting their elephant tusks, or rhino horns, or catching an exotic parrot, are more likely to do that. So we are connected with animals and their conservation. If we help people in Africa, South Asia, South America, like, we help animals, and it's kind of like a reciprocal, one benefits the other. And sadly, only since I was in undergrad and when I was finishing undergrad, that people started realizing that these were interconnected. Which, we're one world, so of course we're going to be connected. One thing, what I do to some person eventually might affect Jack or you. We won't necessarily know it, but it does. We do affect each other, and we affect animals, and they affect us. I mean, it's like ecosystem services. Basically, animals that serve a benefit to stabilizing the ecosystem that we benefit off of. So we, again, I'm going back to that word. The animals are in our charge. Kind of almost sometimes in a bigger scale than that.
It's true. And I think, a lot of times conservation isn't just about conserving the species. It's about conserving the species through education, through educating the population where that species lives. And, so a lot of times when people donate to a conservation program, it's, oh, well, I'm just, I'm saving that bird, or I'm saving that mammal, whatever that mammal may be. But it's not necessarily just that one species. It's, maybe that is a keystone species for a whole ecosystem. Maybe you can look and see what's going on, and how does my supporting conservation affect the bigger picture? And I think, we start with the work that you're doing, Taylor. And, I would love to have people who are listening, support what you're doing and help you with what you're doing. And then that will expand to the bigger picture, I think. So how can people support your work, Taylor, aside from samples, or how can they help you?
Samples is the biggest thing. Samples really is the biggest thing, because if I don't have samples of a population I want to manage, I really can't do anything. But it's also taking down records of, keeping records of your birds, breeding, when the birds first started breeding, what the hatch date was, getting it sexed, which, if someone wants to collaborate with me on, and I consider any breeder, pet owner that wants to send their samples as a collaborator, as an equal. It's not me telling you what to do necessarily. It's us working together on this project. I have a particular skill set and a particular knowledge base, but also any one of our viewers right now has that. So you can go to the website. It's in construction-ish right now, so hopefully by this end of this weekend, in the next week, it will be complete. But you can always email me at this address, or take that same part before the @ and just put uchicago.edu or at fieldmuseum.org. Preferably this one, so I can keep track of captive, the population management. But yeah, if you are anyone's interested in submitting samples, helping to make better breeding records for their flocks, their individual flocks, the biggest thing is participation in more collaborative breeding between different. Ideally, we start with the captive breeders, and then we try and expand into zoos for those 8 species that there are SSPs, but then potentially creating more and showing to the zoo community that there are people that, privately own birds that want to contribute in conservation in a meaningful way that actually helps them. So those are the best ways potentially for people to help out. And, donations to the Field Museum or to Terra Wildlife Genomics for funding, obviously. Most of the time, I'm pretty good at monies, but more money helps out, makes a difference between us doing a reduced representation sequencing versus a whole genome sequencing. So species right now that I'm trying to start up. So we have the blue-headed macaws, obviously. So if anyone else that has blue-headed macaws, please reach out, because the one shortcoming of that project was that we only had 2 aviaries. We need more individuals, show that we have potentially have more genetic diversity, so we can manage this, because AZA does not manage blue-headed macaws. The next tier of projects is the blue-throated macaw, the hyacinth macaw, the golden conure,
Did you see,
and potentially
Taylor, did you
Susan's
see that Dr.
message?
would like to work with you?
I actually know who has her samples, so I've been trying to get them from him for actually a couple weeks now. So I got you, Susan. I got you. I am that bad boyfriend you can't get rid of, that I keep. But instead of asking for your phone number, I keep on asking for blood samples or sequence data. I am that type of person. Yeah, no, maybe people don't want to talk to me after that point, but I'm trying to figure that out. But also the blue and butterflies fly by. But also the blue-throated conure and the gray-breasted conure, I'm also trying to start projects for. I have contacts with the several keepers in Europe, and for the gray-breasted, I have contacts that potentially have all the wild individuals for the gray-breasted, which again, that was 350 to 400 individuals. So trying to actually compare wild versus captive. Interestingly, there might be a chromosomal difference as well. Might be, quotation marks. But at least on a regular genetic basis, no one's really kind of done that work before. People are now starting to do that. So if anyone wants to submit samples, that would be, and support these type of projects, that would be the biggest thing.
All right. And then Adrian just
And
asked, are you softbills and finches?
Yep. Yes, as well. There are other people that have done passerines. Personally, I kind of despise passerines because everyone studies passerines. Okay, so passerines, for anyone that doesn't know, are perching birds. Everyone studies passerines. Everyone told me to work on my dissertation on passerines instead of parrots. However, like this type of stuff can be applied to any group whatsoever. So I initially wanted to start with parrots, obviously, because I love parrots. I have a parrot. But I want to work on species that are within the pet trade in general. So going to finches, other random things. People own alligators as pets. People own, anacondas as pets. People own, random lizards as pets and frogs. I want to expand it to that eventually, after getting the pipelines and everything down.
Okay. And if you live in Texas like Jack, you can own pretty much anything. So
Yeah, some people have told me I should move to Texas, and I'm just like, the politics makes me maybe go no. But the fact that like open terrain and you can almost own anything, and there's a lot of breeders there from all sorts of species, makes me kind of want to go yes, or maybe make like a couple month excursion down to Texas every once in a while.
there it
But I
is.
have to be wealthy. I have to make money for that.
So I think that's all the time we have, right, Jack? Are you?
Yeah, I think this was a great session with a ton of information, and I think the really interesting part of this is no matter where you are in the avicultural community, if you are working in zoos, if you are a breeder, if you are just a companion pet owner, there is an aspect of this that is applicable to everyone. So I think that's absolutely incredible that you are studying this, that you are furthering our understanding of this, and reaching out to people. Looking through the chat, we had a ton of people who are willing to reach out and to help you with that research, because I think we all benefit from having a better understanding of our birds. So
Absolutely. And
I love
anytime,
you all.
and we will definitely continue, Taylor, to get the word out there for you. And this is a great group of people that we've got listening, and they are pretty cool about all aspects of aviculture. So I want to thank you for joining us. And I learned a lot, and I know these guys did too. So Jack, do we have a trivia question tonight?
we do have a trivia question. So if you guys are just tuning in, remember, we have a trivia question every single week. We give away something every single week. So make sure you don't miss a Flock Talk, because you have to be live to answer the question to get your prize. And our trivia question tonight, what are the avian sex chromosomes?
Taylor,
So
you
we
can't
actually,
win.
yeah, you can't win.
I know.
If you told us
That's
the
why I'm covering my mouth.
answer.
I would.
Well,
My habit would be to just answer the question, and then that ruins it for everyone else. So I'm covering my mouth.
when we have vets come out to work on our animals, they always bring vet students and they ask them questions, and I always answer them. And one time the vet was like, "Okay, I'm glad you know the answer, but you need to let them answer." So this was actually a question that we covered the uh, in the presentation.
So
That was close.
close. We let Nick decide too.
No. Close again.
I
Yes.
think.
All right. So Iris, we now owe Iris 2 toys, I believe. Or actually we owe her 1. And Iris, we can do a consult. Jack and I can do a consult with you guys if you're interested. So just reach out and let us know. We go back and forth between sometimes it's a toy, sometimes it's a little parrot tchotchke, for lack of a better term. Sometimes it's a consult. So you never know. So you should pay attention every single week. Because you never know. It could be $1 million. No, not really.
I don't have $1 million to give them.
Jack, it could get people to listen.
They're going to want $1 million I don't have.
I said it could be.
Oh, okay.
So
so we need to come up with $1 million, Robin?
No.
Because
No.
that sounds like a good segue to talk about what our presentation is for next week.
What's our presentation for next week, Jack?
So next week, we are going to be turning tricks.
Well, hello there.
We are going to be turning tricks
Okay, that
into
makes-
husbandry behaviors.
So,
it's not
wait,
as
turning
exciting
tricks
as it
into
might
husbands?
seem.
Turning tricks into husbands? Is that what we're talking about?
I don't know.
Yeah.
Jack, did you come up with that title all by yourself? Because it wasn't me. All right.
It's a fun
Well,
title.
It is a fun title. So we will see you guys. Thank you again, Taylor. It was amazing information. And I know that if you look through the comments, people are super excited about learning more about this. So thank you very much. And I'm glad we were all at AFA.
Thank you for having me.
And we will see you all next week for, I'm not even going to say it again. Candy wants a check for that $1 million. So turning tricks.
Well, hello there.
It's not as good when I say it, Jack. All right, guys, we'll see you later. Take care. Have a good week and stay safe.